Hello, everyone. It's our honor having with us today Professor DEDON from MIT. Professor DEDON is currently Singapore professor at the Department of Biological Engineering at MIT. He is lead principal investigator in the Singapore MIT Alliance Research and Technology in the program of Antimicrobial Drug Resistance Program. And Pete is also a founder of the best university in Bangkok. That is what I learned during my last visit a few weeks ago in Bangkok, the Chulalongkorn Graduate Institute, right? Yeah. Pete is a fellow of the American Chemical Society, and he is also a fellow of the American Academy of Arts and Science. Pete has commercialized his technology and discovery in several biotech startup companies. Pete today will talk about his research program in applying a chemical approach to understand nucleic acid biology. In particular, so he will introduce a part of his work in developing analytical and informative tools for discovering and quantitative analysis of epigenetic and transcriptomic infectious disease and cancers. Yeah, sorry, I have to read because it's pretty much different from what I was in your lab back to many years ago. It's pretty much very different. Sorry. Yeah. Today, Pete's talk is rethinking the central dogma in the age of epigenomes and epitranscriptomes. New biology needs new tools. I think that's mass spectrometry, right? Yeah, and I also learned a new term in here, dogma. Pete said is unbreakable principle, right? Unbreakable rules. Yeah. Okay, without further ado, please join me to welcome Pete.
Can you hear me? There, it's working. Well, thank you. Thank you very much, Simon. It's nice to see you again. It's nice to be back. Nice to see some friends that I've seen. It was 5 years ago, we were just thinking, when the last time I was here. So this is very enjoyable to come back in. And yes, the lab has changed a lot since you left. That was 152 years ago, wasn't it? Something like that, right? So I changed the title on you, mainly because I wanted to talk both about epigenetics. This is the disclosures. I'm obliged to tell you some of the things I'll talk about could benefit me personally in the companies that I own and all of that kind of thing. Tom Bagley is a longtime collaborator, 15 years. I'll show you a lot of the things we've done together. So I have the two groups, one at MIT and one in Singapore. They're one lab. We have the same instruments. We do much the same work. We have our group meeting together every week. And constantly evolving with students and postdocs, but both very wonderful students and staff. So the central dogma idea here that goes back to biology from high school, right? You have genes. Genes get transcribed into RNA. The RNA gets transcribed into proteins, and that's life. All right? This is supposedly the inviolate dogmatic rule. And when some of the Nobel Prize winners hear that I'm talking about alternative genetic codes, I get a lot of grief. I get into a lot of trouble when I say things like that. You know, if you look now at the human genome, it's about 20,000 genes, okay? What tells when to turn a gene on? And my biology friends will all say, well, it's the transcription factors. Well, what told the transcription factors to turn on? All right, this is where the epigenomes and the epitranscriptomes come into play. The epigenome being the DNA modifications, and in humans, the histone protein modifications that regulate transcription. It's the scheduler for all transcription that takes place. The epitranscriptome is much newer. It's all the modifications on all forms of RNA, and it regulates translation. So you're scheduling gene expression at the level of transcription with the epigenome and at the level of translation with the epitranscriptome. The genes themselves is a catalog. They don't know when to turn on and off. So the determining how much protein and when to make it is entirely due to epigenomes and epitranscriptomes. Now, I want to first talk about the epigenome. I'm purposefully not going to talk about some things that I'm going to talk about tomorrow at the Hong Kong Society of Mass Spectrometry. I didn't want to be repetitive and boring. I didn't want Simon falling asleep tomorrow during my talk. So I'll focus on a few different things. I want to talk about my epigenetics work today, which has blossomed in the last few years. So I work with another professor at MIT. His name is Eric Alm. He's a computational biologist and a microbiome specialist. His group of people working on the microbiome and my group, this portion of my group working on this. What we're going to talk about here is sulfur modifications of DNA, phosphorothioates. Every chemist in here is probably very familiar with this idea. And I'll tell the story of Fritz Eckstein, who invented the chemistry. I went to his 90th birthday in Göttingen, Germany, last fall to celebrate phosphorothioates. And I'm the natural products guy. But what we have are these phosphorothioate epigenetic marks are now representing about 20% of your gut microbiome has bacteria with these modifications. What are they doing? Where do they go? And how are they affected by human disease? So I'm a medical doctor. I have an MD degree. I have a PhD in pharmacology. I'm a nucleic acid chemist. So I'm always looking for ways to consider how my science affects human disease, looking for ways to translate the science to something practical. And this one, I'll end this section with a clinical study that we're starting now. So this goes back to a Chinese graduate student at that time at Shanghai Jiaotang University, Wang Lianrong, who came to my lab, said, I want to study how sulfur gets into DNA. And I had never heard of this. And I said, well, it sounds very cool. I don't have any money to pay you. That's okay. I will work for free. My husband's a postdoc over at Harvard, and he has income. And I said, sure, fine. Who wouldn't turn down somebody who wants to work for free? Well, what she ended up discovering was that sulfur gets incorporated into DNA in the non-bridging oxygen in the phosphate backbone as a phosphorothioate. And here comes Fritz Eckstein. He had invented this as a synthetic technology 30 years earlier. All oligonucleotides now used commercially, therapeutically, have phosphothioates in them. They prevent degradation by nucleases when you're delivering this compound into the blood and getting it to the cells to do its therapeutic outcome. So all nucleic acid, messenger RNA therapeutics have phosphothioates. So his invention was celebrated at his 90th birthday with all kinds of industrial people on hand. I was the only natural products person. We discovered them as an enzymatic modification of DNA in bacteria. Fritz wrote the news and views that accompanied our original paper back in 2007. Lianrong is now a very senior professor at Wuhan University, still working in this area. And it goes back even further to Ding Zhexin, who is a very senior person now at Wuhan and Shanghai Jiaotang. He saw, when they ran tryst gels in the early days, degradation, and he worked it out that there was sulfur getting into the DNA. They didn't know the structure. And they could tell the genetics. They've gone through and worked out all the genetics. And so Lianrong came to the lab and showed that sulfur was getting incorporated as a phosphorothioate by a series of enzymes, DND A through E, put the modification into the DNA. It's part of a restriction modification system in bacteria to protect against phages and attacking. And so there's a restriction system, DND F through H. And Zhexin and a whole team now, De Linyu, have worked out some of the biology pretty well, but we still don't know the enzymology. It isn't all that clear. So here's the general pattern here. You've got a cluster of these 5 genes that go through a series of activation steps. You have to put a strand break in, activate with an adenosine activation, and then there's a sulfur transferase activity, very poorly defined. And finally, you end up making the phosphorothioate in the DNA. It's put in after the DNA is made, and it's I'll show you the interesting part of the biology. The restriction system is also very interesting in that it isn't clear how the enzymes monitor for the presence of the phosphorothioate. Not all the consensus sequences are modified. So how does the cell know not to cut at one place when there isn't a phosphorothioate? It's a very interesting system. So Lianrong also discovered another system called SSPE that actually is activated by sequence-specific phosphorothioates, and it nicks the bacteriophage. When the bacteriophage virus infects the cell, it doesn't have phosphorothioates, and it goes and destroys the RNA. It's a classic restriction modification system. But it's turning out that there are many of these systems all through the bacterial world, all over the place, and that's what is the exciting part here. All right, so where we came in now to start to translate this is to develop a mass spec-based method. Simply isolate the DNA, hydrolyze it to nucleosides, and fortunately, if you use nuclease P1, it can't digest a phosphorothioate. So you have a limit digest of nucleosides and then dinucleotides, just like this, bridged by a phosphorothioate. So we synthesized chemically, and you may remember Stephanie Kellner. We ordered from a German company all the possible dinucleotide combinations, and they resolve very nicely on reverse phase HPLC, and you can get a beautiful LCMS, very sensitive analysis of all possible dinucleotides. So now we digest the DNA and look for these dinucleotides to tell if a bacterium has the phosphorothioate. And we did a survey paper that we published in PNAS, again, quite a long time ago, looking at hundreds of different kinds of bacteria. All over the place. And you see these 300 to 500 per 10 to the sixth down to a few mistakes. These little 1 per 10 to the sixth, the enzymes are kind of sloppy. They'll put phosphorothioates in the wrong places because they're shape sensors. I'll show you some of that data in a minute. But this covers almost all bacterial genuses and species have a strain that contains D&D genes and phosphothioates, or SSP genes. Every one. It's horizontally transferred. It's a wonderful restriction modification system. Your human gut microbiome, as I'll show you in a minute, 20% of the bugs have it present. So at the same time we're developing mass spec tools, we also want to develop sequencing tools. How do we map phosphorothioates in a genome and exploit the chemical sensitivity? Well, Fritz Eckstein, again, going back to the 1980s, had discovered that simple iodine treatment puts a strand break wherever there's a phosphorothioate. So we could take advantage of that by blocking all the existing strand breaks, then putting in iodine treatment to put a break where there's a phosphorothioate, and now you map that using enzymes to do standard DNA sequencing. And we're able then to localize at single nucleotide resolution the locations of the phosphorothioates. We could do the same thing with PAC Bioscience, single molecule real-time sequencing. We published a paper doing that with another Chinese student, Cao Bo, back about 10 years ago. So we have a bunch of tools now to measure the modifications and then sequence them. And when you're sequencing, you then also identify the bacterium. We'll exploit that to do microbiome work coming up. So this is a Circos plot. All the green dashes are phosphorothioates along the chromosome in E. coli bacteria. That's about 1 per 10 to the third nucleotides has a phosphorothioate present. Here's M6A, another epigenetic mark in that bacteria. This is PAC Bioscience, single molecule real-time sequencing. And there's a phosphorothioate, GAAC, GTTC, very common motif in bacteria like E. coli. Problem was, only 10% of those GAAC sites had a phosphorothioate. The other 90% were not modified. Why aren't the restriction enzymes cutting at the unmodified site? Well, it's a mystery that we've solved pretty recently, where the enzymes are sensing shape. They'll go after a specific shape of DNA, not necessarily a specific sequence, and the cell maintains a certain density of phosphorothioates. It's not the absolute number, it's the number per 10 to the sixth. And we discovered this, and I'll show you in 1 second here. But this is Remo Rose at University of Southern California, has a computational tool to look at DNA sequences and identify parameters of shape. And when he did that, he found very specific shapes for phosphorothioate modified DNA sequences, and they were predictable. And the enzymes, each different enzyme from each different bacteria senses a different shape, and that shape is the consensus sequence. And so for these things, it's not one strict sequence. It's some related sequences that kind of look alike. They have the same shape. And there's an interesting problem that in some bugs, when there's an M6A present at the consensus sequence, if there's 3 or 4 consensus sequences in one bacteria, if there's another modification there, the phosphorothioate can't put it in. And the number of phosphorothioates doesn't go down when we change the number of M6As. It stays constant. So all we do is get them to shift from one consensus sequence to the number, but the density is always the same. So the bacteria, we think, the enzymes, are somehow sensing density of modifications. And if the density is right, they don't cut. But if the density is wrong, like a bacteriophage that has 0, then they cut. So now we want to take our sequencing tools and apply it now to figure out where these restriction sites are. Okay, so most recently now, and the more exciting part of this is looking at the human microbiome. Thousands of bacteria in your gut, very popular now. The population of bacteria in everybody's gut is different. And from one culture to the next, it's different. It changes with your diet. And you're kind of locked in as a child to your microbiome. And you keep it roughly throughout your adult life unless you shift continents and completely change your environment. And then there are shifts that take place. And of course, you're all pretty familiar now with the news headlines about the metabolites in the gut microbiome becoming essential chemicals in the human body. And in fact, there's a tRNA modification that we study called QSine that humans can't make. And they have to get it from diet and microbiome. And it turns out your microbiome is like the major source for the precursor. And there are now associations between microbiomes and psychopathology, that the cueing is required for normal brain function, and that by changing your microbiome, you can actually change your psychological profile. So very amazing connections between the microbiome chemistry, biochemistry, and human behavior. Well, so here I come in to ask now, how do the microbiomes that we're going to see affect human health? Do the gut bacteria have them? Which bacteria have them? And it's oxidizable sulfur, right? You're chemists. You could probably recognize that the sulfur is nucleophilic and very easily oxidized. And so we set out to test this now with Eric, who has started a company called Open Biome, where they get fecal samples from healthy people, and we extract the DNA and look for the phosphorothioates. He's also cultured 20,000 different strains of bacteria from healthy human gut microbiome. Many of them are anaerobic, many of them are fussy, but they've cultured them and they now have a library that they've fully sequenced all the genomes. So we took advantage of that to go do genomics and analyze which ones have D&D genes, SSP genes. We did our mass spec analysis on fecal DNA and the sequencing of that DNA with our techniques to now get a big picture of how many bacteria in your gut have this phosphorothioate, which bacteria, what's the consensus sequence, what's the gene that's putting it in. And you do have to get comfortable with poop when you're in the microbiome world, and it's disgusting, and you just learn to talk about it without getting upset. But we will extract the DNA. We've had to do some technical adjustments. I'll show you in a minute. Do our mass spec, do the sequencing, and then do big data analysis to discover what is going on with people. So this goes back to classic analytical chemistry. How do you extract the DNA from bacteria in fecal matter? The kits that were out there, like QIAGEN kits, probably some of you have used those for isolating DNA, they're horrible at getting DNA out of feces. And we had to go use an optimized protocol to get a much higher yield of DNA, because you don't get a lot of poop from these banks. You only get very small samples. So we had to completely re-optimize the extraction strategy. Classic analytical chemistry. Sample quality is very important. Now, we started with mice. And this is where Stephanie Kellner did the first study. And we collected feces from cage mates of mice. And mice are amazingly consistent. Both males and females have exactly the same phosphorothioate spectrum. All the mice have exactly the same thing. And here we're collecting about 11 out of the 16 possible dinucleotide combinations. All the mice in this laboratory had exactly the same spectrum, some for very abundant modifications, some less abundant modifications. Males and females were pretty similar. Well, mice are coprophagic. They eat poop. And they all live in the same cage, so they're all sharing exactly the same microbiome. They're all highly inbred genetically. So you would expect it. Mice are going to have all the same microbiomes. Humans do not. Humans do not eat much poop, and we're all very genetically diverse, and we're all very different. And so these are 1, 2, 3. This is a bunch of people. Each different color is a different donor, and they're all over the map. We've now got some people in which we found 0 phosphorothioates, undetectable. They may be there, but below the limit of detection of the mass spec. But in this case, again, we're seeing about 11 out of 16 possible dinucleotides present here. Are there some dinucleotides that just don't exist in nature? Some sequence context, we don't know yet. And now how do they change as a function? Oh, here's the comparison of mice and humans. Very similar, except for these ones. Neither humans nor mice have a TA or TT dinucleotide. We don't know why. This is just Wild West exploring. So now, how do these behave in people? How do they change from day to day? How do they change with your diet? And one of the most interesting things we discovered, these are 4 different people, 4 different graphs, and it's a time course. This one's the most granular time course, going for an entire year, maybe every week or so for a year. Here we have some shorter time courses, but what do you see? Down, up, up, down. It's almost sinusoidal variation in the abundance of the dinucleotides. The spectrum does not change. In this person, they have 4, and they always have 4, and they will always have 4 forever. But the proportions of the 4 stay the same, but the total amount goes up and down. This is more variation than you'd expect in the microbiome in a person. So there's something very cyclic happening here in these people's diet, behavior. The abundance of these bacteria is shifting, in some cases, 5 or 10-fold. That's huge variation in sulfur-containing bacteria in the gut. We have no idea why. We're submitting a big grant application now to figure out what's happening. We don't have the sequences for these bacteria. We're now sequencing the fecal DNA using that phosphorothioate sequencing trick. So we can both identify the bacteria and then look at the consensus sequence and ask which bacteria. Here, this person's got 3 dinucleotides. This person only has 2. This person's got 3 and 4. So everybody's different in the spectrum of bacteria that contain these, and they're varying cyclically almost. Everybody's got a different periodicity. There isn't one periodicity for everybody. So we don't know if it's exercise. Is this when Pete DEDON was yelling at this volunteer in the lab and they got distressed and then they went back up when the experiment worked? We don't know. We have no idea why these things are varying, but it's very fun to watch this. So here now we want to be able to ask some different questions. We want to be able to go look at all the sequenced genomes for 20,000 gut microbiome bacteria and start to ask which ones have D&D genes, which ones have SSP genes, and go dig. And again, this is a fishing expedition. We have no hypothesis. We don't know what we're looking for. The data is available. I have several computational biologists in the lab now who can now go mine and discover and search. And so we developed algorithms to go searching through the genomes and discover now what we think are the genes putting the phosphorothioates in, and then we go confirm it with mass spec. We'll take those isolates that we have looked at their sequence genome, we'll measure the PTs, and confirm that they have these things in there. And so here we have these microbiome libraries that I told you about, the 3,000 isolates at the Broad, the global microbiome library. So a total of about 20,000, this is an old slide actually, and it's about 20,000 sequenced genomes of isolated bacteria from gut microbes. Very powerful system. Okay, and when we do that, here's all the different genuses of bacteria. And the size of the circle tells you the abundance of those that have D&D genes. There's SSP genes that I told you about. Well, what the student discovered was there's a new phosphorothioate modification called BREX. And who studies BREX? I don't know. So you go search the literature and you find out that there's a woman at the University of Chicago who studies BREX, the BREX family of, it's known to be a, where did I put that note? It's known to be a phage defense mechanism, but no idea that it was a phosphorothioate. We got the strains of bacteria from her, and sure enough, they have phosphorothioates, but they're huge. This BREX4, this is in your gut microbiome. So that all your bacteria have a variety of genetic systems to put the phosphorothioates in, and there's more out there. The student who did this, Yifeng Yuan, says that there's evidence for 3 or 4 more systems. He doesn't have the statistical power yet to prove that they exist. Plus, he has to get out of my lab and go get a real job at some point here. So, yeah, and here we go, looking at the phosphorothioates now in some Brex mutants. The point being now, the Brex is a very simple system. Only 2 enzymes are needed to put the phosphorothioates in, compared to 4 for SSP and D&D. So, and they're 2 very different enzymes. So the biochemistry of putting these things in is highly variant and different mechanistically. And yet it's conserved in all these bacteria to have these phosphorothioates present. Okay, so now we go through and look at the PT landscape in the fecal DNA, apply our sequencing tools. And we actually had to go change our sequencing technology. When you do fecal DNA, it's not like taking a pure culture of bacteria, right? Where all the DNA is exactly the same in every bacterium. When you take fecal DNA out, you've got thousands of bacteria in different proportions. So when you go apply sequencing technology, if it's noisy, you lose the specificity and sensitivity of identifying any one bacterium, because your signal is now 0.1% of what it used to be. Classic analytical problem, right? When signal drops below noise, we're in trouble. So we had to lower the noise and improve the sensitivity. And by doing some tweaks, we were able, just chemical tweaks on the sequencing thing, we were able to get a 10 to 20-fold increase in sensitivity. So instead of 5 reads on the sequencer giving us the identity of something, which is terrible, we're now up to like 50 or 100. We get big stacks of data to tell us where the sequences are, what bacteria they're from. And so here from the human gut microbiome, again, here's all the different genuses that are there. Here's the big one, firmicutes, things like Bacteroides. I know Young is studying Bacteroides now. The 800-pound gorillas of phosphorothioate-containing bacteria are these few Bacteroides, firmicutes, and a few other strains. But then there are other ones, Proteobacteria, I can't even say the names of some of these bacteria, showing you the dinucleotides that are present based on the sequence specificity, the motifs that we identified, the frequency. So highly abundant bacteria now, and mainly the Bacteroides and firmicutes. So we're mapping the landscape here. We don't know what or why these things are the way they are. But in everybody we're studying, it's always firmicutes and Bacteroides. And are those the ones that are undulating up and down? And it's only a subset of the Bacteroides. There's hundreds of different strains of Bacteroides in the gut microbiome. This just repeats the same thing, shows you the same data. I won't harp on that. The big point now for me is, all right, you got all these bacteria in your gut. And all these bacteria now have a redox-sensitive sulfur that could get alkylated and cause genetic damage. And they're sitting in a sea of inflammatory chemicals when you have inflammatory bowel disease, Crohn's, ulcerative colitis. I got my start at MIT doing chemistry of the innate immune system and how it damages DNA. Nitric oxide, hypochlorous acid, carbonate radical anion, all these super oxidizing species are now in the gut, and your bacteria are swimming in this. Is there something that the sulfur does that makes the cells different? Well, this is Stephanie Kellner. Was she in the lab when you were there?
I left already.
You had left already, and then she came. All right. She came in and demonstrated, the bottom line is, bacteria that have a phosphorothioate are exquisitely more sensitive to hypochlorous acid made by neutrophils. If you simply get rid of the phosphorothioate by knocking the D&D gene out, they're 1,000 times less sensitive to killing by neutrophils. So for us, this led to a simple-minded hypothesis. If we have a big raging inflammation with tons of neutrophils, the population of phosphorothioate-containing bacteria is going to drop. That was my simple-minded hypothesis. Well, and then we met a real IBD doc. I'm not a practicing MD. And so he's Josh Korzenik, heads the Brigham and Women's Hospital Crohn's Disease Center. He had a different hypothesis. He said when you have inflammation in the gut, you slough off the mucosa. Well, what's in mucus covering of your intestine? It's all this sulfur-rich proteoglycans, all these complicated heparin-like molecules that get sloughed off and they go into the goop of the bacteria. He said the bacteria are probably going to have a bonanza. It's like a buffet of sulfur, and all the sulfur-utilizing bacteria will thrive and they'll grow like mad. So we have these opposite hypotheses. And so what did we do? We collaborated with him, and he gave us samples of fecal DNA, 15 from Crohn's disease, and then we have people who have a flare-up of Crohn's disease. We have people who have Crohn's disease, but they're inactive. Same thing with ulcerative colitis. We then took our DNA isolation method, and we quantified the phosphorothioates in all these people. And here's the controls, healthy people, right? It's fairly low levels, about what we saw previously. But then look at the people with Crohn's, either inactive or active Crohn's disease. There's a couple of them with levels that are some 200 times higher than the healthy patients. Here it is. So I think Josh Korzenik's hypothesis was right, that some of these bacteria are just swimming in a sea of sulfur. They're eating it up. They're thriving and having an advantage. But not always, not some of them. Some of the people, this is the first time we've ever seen humans who lacked detectable phosphorothioates. That's a new one. Do they not have any bacteria in their gut? We're collaborating with an Irish group who says that in diet, the cuine content in diet, that there are British people who can have a diet with 0 cuine in it. And if you have a gut microbiome that lacks the bacteria that could make cuine, you're in trouble. So here we have people lacking bacteria that make phosphorothioates. That just tells you the diversity that's out there. Ulcerative colitis, a little more complicated. But these people apparently were actively taking drugs for their disease while they were collecting the donation. Here, they weren't actively taking drugs necessarily. So we don't have any definitive answers yet, but we're going to expand this study. But it looks like people with Crohn's disease have a very different population, a big blooming of phosphorothioate-containing bacteria. So how do you translate that into something as a therapy? Do we want to give a therapeutic that decreases the number of phosphorothioate-containing bacteria? Can I make an antibiotic that targets phosphothioate bacteria? Is this the cause of some of the symptoms of Crohn's disease? That one we don't know either. So if I make an antibiotic that only kills phosphothioate-containing bacteria, could I relieve the symptoms of people with Crohn's disease? Don't know. We'll see. That's the hypothesis. This just shows you the average, the spread in the data from 11 donors, 123 samplings from 11 donors. You see that the spread is pretty big. Here's the controls over here, which is a little lower than our larger population. But still, even with broad sampling of controls, the ulcerative colitis people have some real big outliers in that front. Just to brag a little bit, we've done a whole bunch of other discovery. We do a lot of mass spec-based discovery of unusual DNA modifications. And here's one. This is the QSC I was talking about. It's in the wobble position of tRNA, which I won't get a chance to talk much about today. 7-deazoguanine. Very strange thing. It's made from GTP in the cytosol. Then there's a transglycosylase that pops the normal guanine out and pops the 7-deazog in. Well, it's happening in DNA, too. These are all the structures we've recently discovered of 7-deazoguanine modifications in DNA. And interestingly, in some bacteriophage, 100% of the guanines are substituted with one of these. They actually make the trinucleotide, and the polymerase puts it in as if it were G. It's a strange world of epigenetics out there. And this is just the tip of the iceberg on weird DNA modifications. These ones are in your gut microbiome. About 5% of the gut microbiome has one of these 7-deazogs in it. Very bizarre stuff. Okay, we're at 45 minutes. and I don't want you to sit here and go to sleep. So I'm going to move through some of this, and I'm going to skip some. I always put too many slides in. There's too much fun going on. I did want to switch gears for a moment and talk about the RNA modifications, the Epitranscriptome. This is an amazing world. Eye candy here. It's a picture of the 170 known modifications from the Modomics website. These are all the chemical structures that have been defined in viruses, bacteria, parasites, humans, yeast. 170 different chemical structures. Bizarre, exotic things, isopentenyl, adenosine, this long isopentenyl structure. We saw the cuine, 7-deazoguanine with a glycosylation on it. All organisms have about 50 different modifications. Humans have about 52. And it just depends. Bacteria have about 45. But every organism has them. They're in every form of RNA in every organism. Your microRNAs, your messenger RNA, your tRNA, especially tRNA, which is what I'll talk about, have all of these modifications in them. This is the new world. This is not in Harvey Lotus's textbook on cell biology. It's not out there yet. It's only about 5 to 10 years old that these things are actually very critical players in the function of the RNA. Now, tRNA is where I'm the most interested. Typical tRNA, 80 to 90 nucleotides long. 10% of those nucleotides are something other than A, G, C, and U, which is what we learn in bio class, right? This orange dots here are sites of conserved modifications in tRNAs across all organisms. Here's the anticodon, right, that reads the codon in the messenger RNA. The wobble position is the most heavily modified, this is the wobble position, is the most heavily modified, position 37, 36, 35. I think the numbering is wrong on this. I think this ought to be 1 over. Anyway, that's the wobble position. Most heavily modified one, and this regulates how the tRNA chooses its synonymous codon. Remember, every amino acid, most of them have more than 1 codon. Some of them have 6 codons. And not every tRNA will read those codons until they're modified. These modifications function in all manner of biosynthesis of tRNA, regulating translation. Well, here's what Tom Bagley and I have discovered. He used to be a postdoc at MIT. He left MIT, took a faculty position, asked me to be on his advisory board because I did mass spec, and he was studying how tRNA modifications regulated the stress response in yeast. And I went to the first meeting as his advisor, saw what he was doing, and he measured one modification using radioactivity. And I said, aren't there like 50 or 100 of those stupid things? And I said, we can measure them all on the mass spec. So I resigned from his committee that day. We've been collaborating ever since now for about 15 years. And this is what we've done. We've taken and done a lot of omics, mass spec-based omics, genomics, sequencing, and we surveyed all these omes, all the RNAs, all the modifications, all the genomes for codon information, and we came up with this model, which is borne out in a bunch of different publications. When a cell gets stressed, the pool of tRNAs, which in bacteria is about 50, in humans it's about 250, and they have 10% of their nucleotides are modified, those modifications reprogram. They shift around. Some of the wobble modifications come and some of the wobble modifications go. The reprogrammed tRNAs then read their codons better or worse. And it turns out that synonymous codons, this is Tom's discovery now, are non-randomly used in the genome. How do you pick the one of 6 that codes for arginine? It's not random. Now, the old people, sorry old guys, old women in the audience here, they would say, well, there's the optimal codon. Bacteria use one codon over the other. It's not the way it works. What we discovered was that their messenger RNAs from families of stress response genes are enriched with one or the other of the several synonymous codons. The families of messenger RNAs with the enriched codons match the reprogrammed tRNAs for that stress. So a specific stress specifically translates the genes you need to survive that stress. And it's amazing. In fact, we can look at the pattern of modifications in the tRNA, and I can tell you with 85 to 90% certainty what the chemical was that the yeast were exposed to. It's that much information that tells you stress regulates gene expression at the level of translation. That's the epitranscriptome. There's an alternative genetic code, and these things take advantage of it. Now, we've seen this we're just going to publish a paper now on how dengue virus hijacks the epitranscriptome to get its messenger RNA translated. Bacteria, malaria parasites, Peter Pricer, we've got a bunch of papers with him. Rats, humans, we're getting into this in a big way. Now, what I wanted to spend maybe 5 more minutes talking about here, I don't want everybody to go to sleep, is what we've done now in trying to automate this. If I want to do 1 sample, 10 samples, I can get a student or a postdoc to grab some Eppendorf tubes and we can do this manually, right? Extract the RNA, measure the modifications. But what if I want to do 5,000 samples? Okay, too much work. So we've sought to automate everything. And this is where we started out with a project, current postdoc named Jing Sun has developed an automated way to extract RNA from bacteria and then analyze all the modifications in it. And what we did was we used a library of Pseudomonas aeruginosa bacteria in which we have 5,800 mutant strains, each one lacking a specific gene. Young's familiar with this kind of stuff, but this is a separated, isolated library where it's just lots of 96 well plates, each with a different strain. We wanted to ask the question, how do different genes in the genome affect the profile of RNA modifications? She wasn't going to do this by hand, so she came up with a very clever way to automate this. She developed a beads-based tRNA purification system, not unlike QIAGEN kits. Lysing the cells open, purifying the tRNA. She then puts the plates in the after processing with enzymes to hydrolyze to nucleosides, puts them into the plate reader on an Agilent mass spec, quantifies all the modifications. We had a talented undergrad named Leon Fan develop software to process all of this data, take the mass spec data, convert it to the identity of specific modifications. And here's, for example, what you get when you compare a QIAGEN kit in terms of the small RNA purification to Jing's bead-based purification. Super pure tRNA. And we can also get the ribosomal RNA or any other RNA you want in very pure form with a robot. 96 well plates. Who needs grad students anymore, right? We can do it with robots. I'm being totally silly. She then has an LCMS method, high throughput method, using a very short column. She's able to get pretty good resolution. She can get all 50 nucleotides. It was 52 modifications in a 6-minute run. She's now got it down to 3 minutes. So you can have 3 minutes of run, 3 minutes of wash, and you can increase the throughput now to many, many hundreds of samples per day to get all of this done. Okay, so she put all of this together. I won't bore you with some of the details here. Here's what you get in the end. Each tick mark on the x-axis is one of the strains. And the fold change value from the wild type strain is the y-axis. The gray dots are all the modifications, 52 of them, for each strain that didn't change more than 4-fold. The red went up 4-fold, the blue went down 4-fold in that strain of bacteria. Now you put on your computational biology hat, your informatics hat, and start mining the data. And first of all, you go back and validate and say, if we knock out the RNA modifying enzyme, did the modification go away? Yeah, there's a whole bunch of dots down at 0, right? Those are the ones. We validated the method because all of these mutants specific to those modifying enzymes, they lost their modifications. Good, bingo, that's nice. A Pearson correlation plot to show which ones are dependent on the others. This confirms some biology that people knew, that you had to put a pseudouridine in before the next one could happen in certain cases. So all of these correlate, again, validate the biology. Now you do like a self-organizing heat map, multivariate statistics. All the blue clusters, that's where the modification went way down for all these genes. So here's the modifications, IO6A going down. Here are modifications that went way up when we knocked out the genes. So there are whole families of genes that are affecting this IO6 modification. What are they? There's only a few modifying enzymes involved in that, so what are all these other genes? Well, I won't get into the dirty details, but when you start looking at all these networks, the bottom line is the cell senses a stress. It's shorted some kind of nutrient. That nutrient is made by an enzyme. That enzyme makes a byproduct that affects the expression of another gene, And those genes then turn on or turn off tRNA modifying enzymes to reprogram the tRNA pool to allow translation of all the enzymes you need to survive that shortage. So you've got whole networks of genes that regulate survival by making the proteins you need to survive that. It's a beautiful story that she's getting ready to publish now. There are iron-sulfur cluster-dependent tRNA modifying enzymes. Well, what happens when you have a lot of nitric oxide, and bacteria make a lot of nitric oxide? That destroys the iron-sulfur clusters. That shuts off that modification. That turns on a bunch of genes that repair iron-sulfur clusters. Cyclic networks of interacting and sensing, regulating the epitranscriptome and translation. I won't get into this. Tomorrow I want to talk about mass spec-based RNA sequencing. How do you quantify modifications along the length of an RNA? We use a next-generation sequencing trick, but it won't map the modifications. I can quantify only a couple of them. So now we're going to develop a mass spec-based technique that, again, we can take the 96-well plate, take a part of the RNA, and go do the modification analysis, take another part of the RNA, and now go do mass spec sequencing. So we can quantify site-specifically where the modifications are changing in which RNA, which is highly critical if you're going to do patient diagnosis and that kind of thing. All right, I'm going to skip this part because it's the end of the day. This is these are tRNAs, and this is a time course of starvation, and here's the sensitivity. And we can quantify the tRNA in the pool and capture all 250 human tRNAs and start quantifying them using a mass spec trick to do this. All right, where are we going to go with this? Ultimately, we want to start applying this high-throughput automated tool to a whole bunch of different studies. So, for example, the Cancer Cell Line Encyclopedia, 2,000 cell lines, 400 of them have deep proteomics. So if we go in and measure all the RNA modifications and the tRNAs, can we now define how the epitranscriptome is screwed up in different forms of cancer? And we know it's screwed up in many different forms of cancer. Here are all the diseases that are known to have an RNA modifying enzyme defect, either in a subpopulation of the patients that have that disease or as the cause of the disease in the first place. Here's one where people with a high level of metal 1, which puts M7G in a tRNA, very bad prognosis if you have head and neck cancer. So we started a company to design chemical inhibitors of metal 1 to get them back to low metal 1 expression and a healthier prognosis for chemotherapy. A lot of different diseases have this. We've got an antibiotic development program going right now because bacteria have a lot of the modifications that humans don't. Bingo, perfect selectivity for making an antibiotic. All right, and this just shows you, just to brag a little bit for Tom and his codon analytics, oncogenes have exactly the opposite codon usage pattern as tumor suppressor genes, which is why when you mess with a tRNA that reads a specific codon, if it allows translation of oncogenes, it's because of the codon biases in these two opposite behaving genes in cells, big, broad families. It tells you the importance of epitranscriptome reprogramming, codon bias translation, determining the cell fate. You become cancer if we start turning on all the oncogenes, and part of it is mediated by enzymes and codons. Whoop, okay, that's it. And I have to do the acknowledgement, I can't skip that. Great lab at MIT, my alums who've graduated. Oh, Simon, your name's not on there. That's because you graduated 152 years ago, pal. You're not a recent alum anymore. Sorry about that. And very grateful to our funding agencies. Thanks for listening.
Yeah, thank you very much, Pete. Yeah, that's two biology for me. Two biology for me. Luckily, we have some biology people here. So, any questions?
A lot of biology in the afternoon.
Li Shangshan.
Yeah, very interesting work. I have a question about your phosphorus project. The since you know where the DNA bases are modified, what are the genes that are modified?
It's a good one, and I didn't show any of the data. There are biases where the phosphorothioates are located at the center part of the gene, or the coding part. They're not located at the 3' and 5' end of the gene where you regulate the transcription and translation.
Right. Okay.
For some reason, they're a little crowded in there. tRNAs have a higher proportion of phosphothioate modifications, tRNA genes, than other genes. So there's some weird, non-random distributions of these things that may reflect what's the effect of putting a sulfur in the backbone.
There must be some regulation, though, per by
well, how does it affect the melting temperature of the DNA when you want to separate the strands and start transcription or DNA replication?
Right.
These things are very subtle and not worked out, but we're finding evidence that it's non-randomly distributed in different genes and different regions, different geographies of the genome.
Is it possible that under the oxidative stress, that the modified gene will be selectively cleaved?
This is like Cindy.
Down the transcript.
Well, it's like Cindy Burroughs' model, right? Where we used to think adoxoguanine was bad, right? She's found that it regulates transcription when adoxo-Gs and these G quartets get oxidized. So the damage is actually a signal to turn on transcription. Is phosphothioates going to be the same way? It would be great. I could get a lot of Jack's papers and get some really nice publications. I don't know yet. I don't know.
Yeah. Well, that's very interesting. If it's low in regulating the transcription or even the translation of certain gene or the related gene, that would be great.
Yeah, I just need $1 million and 100 more grad students, and we could answer all these questions. So.
Yeah, right. Great. Thank you very much.
Thank you.
Thank you, Professor, for the wonderful talk.
I apologize for not talking about your favorite topic very much, the RNA modification.
Okay. Yeah, I will ask you 2 questions about the DNA part. So first one is, I think for the PT modification, is it possible for us to sequence the cell-free DNA? Because we know that a portion of the cell-free DNA can come from the gut microbiome.
Yes, in theory.
Yeah.
I think possibly using a next-generation sequencing method, the sensitivity might be there to pick up some. But I'm not sure, is there any notion of what percentage of cell-free DNA comes from microbiome? Is it like fractions of a percent, or is it like 1%? Do you know?
I remember that could be a significant portion.
Oh, it's an interesting thought.
Yeah, I think maybe if it's good to sequence the cell-free DNA for the input DNA could be like satisfy your method. I think so for cell-free DNA that could be maybe several nanograms for each sample.
That's a lot.
Yeah. Okay.
Yeah. Especially for the sequencing. Now, by mass spec, I'm not sure we could pick up the signal. I mean, we're parts per billion sensitivity, but a few nanograms of gut microbiome DNA and only 10 to 20% of that has a phosphothioate. It could be sketchy, but sequencing, we could just keep amplifying until we see something.
Yeah. Okay, the second question could be, because when I previously do the mass spec for the DNA modifications, like the 6-methyl-A and the 5-methyl-C, I did a digestion using the alkaline phosphonates. So is it possible for the PT modifications exist in like a DNA virus or DNA from the mammalian cells?
So this is something we've thought about. So humans, we can't find it. There was a paper that came out that said RNA, that there was phosphothioates in RNA in humans and yeast and everything. It turned out to be an artifact. It was a 2-O-methyl RNA modification that was 0.1 Dalton different than a phosphothioate containing modification in RNA. And they didn't do high-resolution mass spec to prove it, and they published the paper with low-res, and it was, And Stephanie Kellner and Sal Bo, they were all out in their faculty positions, and they wrote back and said, Pete, did you see this awful paper that came out? And it was a friend of mine who published the paper. And this is one of those diplomacy games, right? They wanted to go publish a paper to reject this, and I said, if you're going to do it, you've got to be rock solid. You have to be absolute proof, and it has to be nice. You have to be nice about this. And they did. They published a beautiful paper. Stephanie, Cao Bo, one person from my lab, they proved that. But can't find it. Can't find phosphorylated thioates in humans. But there are eukaryotic parasites that eat bacteria. And there's evidence now that they incorporate genes from the bacteria in their genome. So these histolytica, Entamoeba histolytica, seems to have phosphorothioates in its DNA. It's the only known eukaryotic organism that does that. But that doesn't mean you can't put the bacterial genes and get them expressed in yeast or humans. Yeah.
Okay, yeah. Thank you so much. Yeah, thank you.
You're welcome.
Thank you, Professor DEDON, for the very brilliant talk. I'm really quite interested in how this, like the PT bacteria, how they like behavior during the IBDs, and like you actually your data shows that the PT bacteria actually they are flourishing during the IBDs, which most of them could be like Bacteroides and Firmicutes. But there are also previous like studies on IBDs stating that like during the IBDs, the diversity of like these two phylum are actually decreasing, while that the proteobacteria they are increasing. Are these two like controversial conclusions?
No, a couple of factors. So we're going to go back and we're going to take that IBD DNA. We only did the mass spec, so we just know the PT level. Mm-hmm. We know that it's increasing because we measure it as an absolute amount, PTs per 10 to the 6th nucleotides. We don't know what bacteria are there, we'll sequence it. But you're right, we read that paper and we kind of thought, wait a minute, if Bacteroides are the 800-pound gorilla, are they going up? Or is it the proteobacteria, which is a small fraction of PT-containing bacteria, are they just exploding and flourishing like mad? Don't know, we have good hypotheses. It also depends on the number of patients you have. How many patients were selected to do that study to show the blooming of different bacteria and coming and going? There's so much inter-individual variation. The statistics becomes mind-boggling. So we'll see. Maybe it is the proteobacteria that we're seeing here.
Thank you.
When I come back again in 2 years, I want to have an answer for you. So,
thanks so much.
Very impressive talk, as always. Actually, I have a question not related to today's talk, but this is a question I always want to ask. Because you are the founding member of at MIT in the Department of Bioengineering, and you are also the co-founder of several biotech startups. And from around 13 years ago until now, you have witnessed the development, the biotech development in Kendall Square. So actually, Hong Kong is eager to be a biotech hub. So what are the key components for the development or the build the biotech ecosystem?
This is the same reason why I'm in Singapore. The Singapore government wants to bring in lots of foreign institutions to bring in expertise and kind of grow a new area for the country. That's national security, food, computers, traffic flow. And they wanted MIT to come in and bring that entrepreneurial spirit. Well, MIT is 170 years old now, and it took that long. It's like wine, right? It's like growing wine. You have rain and sunlight, and what's the minerals in the soil, and how much slope of the hill is there, and what's in the water that they get. And in the end, you get this idiosyncratic thing that you can't reproduce anywhere, right? So how do you take it out of Cambridge and transplant it? Well, it's generations to do it. But from my experience now, being 30 years at MIT, and sort of seeing that slice of it, what did I do to take advantage of that? Part of it was, I've been talking to Simon about this, risk-taking. The willingness to go out and start a company that 9 out of 10 startups fail. And people lose money. Does everybody get angry at you that their company failed? No, because companies fail all the time. You have to have a real, very risk-tolerant attitude, that, especially for drug development. Now, manufacturing and things, it's lower risk. You can do more biomanufacturing, biotech that way. If it's drugs, most drugs fail. So if you get $10 million here, $10 million there to develop a new drug, the 10th drug is going to make $1 billion. And that pays back everything else up to that point. So I say this because MIT's Technology Licensing Office has a budget of $10 million a year. That's huge. Where does the Institute get the money? From all the royalties, from all the things they started. NUS in Singapore has a $1 million budget, and they're very risk averse. They want lots of rock hard proof. Why? Because they don't have the experience yet, the risk tolerance to say, ooh, if I invest money in patenting that, am I going to get that money back? Well, it took MIT 170 years to get there. And you're right, it is a unique environment. You know, you remember the numbers at MIT? When I started as a faculty, Boston was chartered with the question, what's the economic impact of MIT? We were doing this at dinner the other night, right? And they said, how many companies have been started by MIT students? Not just faculty, but students. How many? And still in existence? 100? 1,000? How many companies do people in the audience think were started by MIT students? 30,000. 30,000 companies employing 2.2 million people around the world. What was their gross annual revenue from all those companies last year, 2022? 100 million? A billion? 10 billion? $2.2 trillion US dollars of revenue. If you put all the companies together into one country, it's the 7th largest economic power on the planet. It's goofy, it's mind-boggling. But that's just a very practical point of, hey, I have this idea, and it could be a medicine, it could be something very practical and useful for public health. You don't have to make billions of dollars, but this idea that if you have an idea, you should lower your risk aversion, find a way to see if you can't start it up. And it's hard. I have no idea how to do business. I'm a professor. I stay out of that. Professors are horrible business people, except your old boss, Tim Liu. He's just brilliant at everything, right? But the point being, just take the dive. Just try it. Fall on your face once in a while, and then start over with something else. And don't get punished for failure. That's one of the things. Students at MIT are allowed to fail a lot. It's okay. Pick yourself up, dust yourself off, go find another thing and do it. And learn and say, that was stupid, wasn't it? Okay, let's go be smarter. So I don't know, all of that blithering, does that answer the question?
Yes, thanks.
Yeah. John, I asked him a similar question. I said, why Boston? He said, because Boston is 300 years old. That means it takes time also.
Well, China's a whole lot older than that.
Yeah,
so,
that's why it is China though, right? Okay, yeah, any more questions? Yeah, maybe I ask a general one, just see if I miss your information. So, do we know where is this, or which species of this microbial have this PT on them?
We're learning. We're trying to get the sequencing down to the point where we can identify the species, because it's hard with sequences, because, you know, actinomycete bacteria, the core genome of actinomycete species is 40%. The other 60% of their genome is different strains. Microbiology is crazy. There's no specificity. You don't know the species you're working with. So we're trying to get there, and crunching those computational numbers in the informatics is there. But it's the Bacteroides, Firmicutes, potentially Proteobacteria. Yeah.
Okay. Thank you. More questions?
It's dinner time.
Tea break. Yeah. If not, please join me to thank Pete again.